All right, I think we are ready to start. Hello, everyone. My name is Haley Rivers, and I am the Kupu AmeriCorps member for the Hawaii Experimental Tropical Forest for this year. So, if you haven't seen the webinars before, this is just here so that our researchers can share their work with the community and so everybody can see a little bit of the work that we're doing in the Hawaii Experimental Tropical Forest and at IPIF, the Institute of Pacific Islands Forestry. A few things before we start. This webinar is being recorded. It will be uploaded to a YouTube playlist. You guys will get an email with the link to that after the webinar. Feel free to toggle between gallery view and speaker view as the webinar continues. Once the webinar begins, we ask that your microphones be muted and your video be shut off just to sort of help with any technical glitches we might experience. Feel free to put any of your questions in the chat. Once the presentation is finished, they'll be answered at the end of the webinar, and once the presentation is over, you should be able to turn your video and your microphone on and ask any questions, or feel free to put them in the chat, whichever you feel more comfortable doing. After the webinar, we will be sending out a feedback form, so please take a few minutes to fill it in. There you'll be able to join the mailing list to learn about any future webinars and, just tell us how we can do better and improve, and we can work together to make it a really wonderful series. With all of that being said, I would like to introduce Ellyn Bitume. She has been in Hawaii for 1.5 years. She is a postdoc at the U.S. Forest Service. She has her PhD in evolutionary biology, and she strives to apply those concepts to biological control practices. So now I will hand it over to her. Go ahead, Ellyn.
All right, thanks, Haley. Well, yeah, thank you everybody for coming and showing an interest in this type of research. Today I'm going to be presenting on weed biocontrol for Hawaiian forests. And basically how the talk's going to go is I'm going to give a background of biological control in general, some specific examples in Hawaii, and then I'm going to talk about Koster's curse, which is the plant that I'm currently working on, and show you some results from some experiments and some collections that we've done. So with that, I'll start by saying that I have two pictures here. One is of a native Hawaiian rainforest, and you can see lots of ferns and the ohia, versus an invasion of Koster's curse, which has like completely taken over the understory there. So just from looking at it, you can see that invasive plants can be quite the problem. Okay, so here's another photo of a very big invasive plant in Hawaii called strawberry guava. Again, this is really just to hammer home the point that invasive plants can form these monotypic stands that completely take over native forests, and then also you see there's a lot of native trees there that are dead. So these invasive plants can displace native species, as well as just taking over, changing soil composition, and all sorts of other issues. I wanted to mention Albizia because I was actually hired to work on Albizia, and then with recent travel restrictions because of COVID, that's why I've switched my project to working on Koster's curse. But Albizia is a huge problem in Hawaii on the Big Island. A lot of people know about it because of the damage that it causes when there's high winds and road blockages and things like that. But it's also a huge problem for native Hawaiian forests. And this slide is just to show that when you have a native ohia forest, most of the dominant native trees are alive. But when you have an Albizia invaded forest, most of the native trees are dead. So it's not just a problem for the public as we know it, again, because of damages and road blockages, but it's also really nasty tree that can really disrupt native ecosystems. So I hope by showing you some of the pictures of the scale of the problem of invasive plants, you would all agree that managers really need all available tools possible to combat invasive species in Hawaii. There's so many different kinds, and these are very vulnerable forest ecosystems. So here I've already talked a little bit about albizia and strawberry guava. You guys probably know a lot about Christmasberry and Himalayan raspberry and ginger. Some of the work we're focusing on now is on a family of melastome plants, which include Clidemia, Myconia, and Tubucina. And I like this slide because I really want to also hammer home the point that it's not just the Forest Service that's working on invasive plants. We have a lot of partners: the National Park Service, University of Hawaii, the Hawaii Department of Ag, the LNR, USGS, Fish and Wildlife, HISC, the Hawaii Invasive Species Council, watershed partnerships, and the Nature Conservancy. So there are a lot of scientists and a lot of people on the ground working really hard to combat invasive species. But some of them we just can't mechanically remove. And so again, we need all of the possible tools that we can get. So this is where classical biological control comes in. And if you've never heard of this, I do want to say the caveat that I focus specifically on plant insect, on weed biocontrol, but you can have biological control in many other animal systems. So biological control is where host-specific, co-evolved natural enemies, otherwise known as agents, from the weed's native range are reunited with the invasive plant in the introduced range. Agents then will hopefully establish and increase in abundance to levels that inflict sufficient damage to target weeds. And just to give you a little bit of example, before I moved to Hawaii, I worked in riparian habitats, the complete opposite of this wet side of the Big Island. And here I worked on two different projects. One is giant reed cane and one was tamarix. And you can see here, like these little dots, these are scale insects, and these are a biocontrol agent that were released. And you can see sort of some of the damage that it does. It forces the plant to grow, this sort of, they call it witch's broom, and it stunts the plants and can interrupt its reproductive potential. And here, it's a little bit blurry, but all of this brown that you see here is normally a bright green shrubby plant that has been completely defoliated by a beetle that was released to control this plant. And after many years, we would see a reduction in the amount of plants here. So biocontrol can work and can be specific. But in this case, I also wanted to mention that biocontrol is supposed to be a sustainable and self-perpetuating and effective control method for non-native weeds. Biological control is not usually supposed to be rapid. It's not going to get rid of your invasive plant in a year. Its goal is also not for complete eradication. It's sort of to restore ecological balance and keep low levels of your invasive plant. So I really like this slide. A lot of people, when I tell people what I do, especially on the helo side, they like to bring up mongoose. And I wanted to bring this up for two reasons. One, to say that it's so great whenever I talk to people here that they seem to care a lot about invasive species, bringing in introduced species, threats to their native ecosystem. I have never worked anywhere where people seem as passionate as they do here about protecting their land. And I think that the public should remain skeptical and should hold scientists to account and make sure that we're all doing the right thing for the land. However, I also wanted to bring up this slide just as sort of clarification point, because the mongoose is well known in Hawaii as an example of an unwise intentional introduction. But what you have to know is that it was introduced in 1883 and there was no science, no review, and no regulation. And this is not representative of biological control. As far as I know, it was a private landowner who was tired of the rats. And so he released the mongoose, and we still have this problem today. So just keep in mind that not everything you hear about intentional introduction is the result of a higher standard of scientific practice. So speaking of that, Hawaii does have a very long history of biological control introductions. The first release of agents or weed enemies was against lantana in 1902. I will say that there have been, we have released several different agents, I think 20 or 30 different agents against lantana in the last 100 years, and we haven't really seen that much success. And this highlights one of the biggest problems with biological control is actually getting your agent to establish. So it's not so much that the agent is going to have some non-target effects, but it's actually getting it to establish and work. That's pretty much the hardest part. So in Hawaii over the last 100 years, you can see that we have released several different kinds of biocontrol agents. So herbivores that attack plants, predators that can attack insects, and parasitoids that can attack both plants and insects. And one thing I wanted to highlight is that there was a huge surge of release of biocontrol agents in the 50s, 60s, and 70s. And while this is Hawaii, that's pretty representative of the United States also, the mainland. But what's important to know is that in the 80s and 90s, the numbers go down a lot. And that's not for lack of trying, that's actually because of an increase in high scientific standards. So we wanted to, everyone realized that it was really important to make sure that everything we were doing was going to be in the best interest of the land and minimize non-target effects. And so the scientific rigor went to a higher standard, and fewer agents are being released. And now it can take many, many years to get from the beginning to the end of a biocontrol program. So the last part that I'll talk about non-target effects here. So what are we talking about when we say non-target effects? Most times, the most controversial part of biological control is I don't want to say fear, but the worry that an agent will attack a non-target plant. You know, in California, I had to drive up to farmers who have huge almond farms and tell them that I wanted to release a tiny wasp that attacked this grass that grew right near their whole livelihood. And I had to be able to tell them that this insect was completely host specific and would not touch their almond trees. So this kind of thing seems to be the biggest fear with the public, and with reason. Before 1944, only about half of the releases that were made were host specific. Then it got a lot better. From the 40s to the 70s, about 3/4 of all releases were host specific in Hawaii. But again, as I tried to illustrate on the graph previously, since 1975, 100% of the introductions in Hawaii have been host specific, and that's over 50 introductions. So again, just to stress the point, the biological control has come a long way. We incorporate evolutionary theory into choosing our agents. We're not only studying the insect, but we're studying population structure, studying the potential for evolution. We're really making sure, as best we can, that there will be no unexpected consequences of releasing a new agent. So, how about some good news? This is a great example of the prickly pear cactus, otherwise known as panini, in Hawaii. This is a photo from the Big Island in 1950. Biocontrols were released during those 49, 50, 51. And 4 years later, this is the same field, and you can see that there are fewer of the cactus, and that you can also see a bunch of skeletons from where the cactus have died. And in 1958, the same field is almost devoid of the cactus. But I want you to notice that that took 8 years. So biological control is a very slow process. And again, you're still going to see some cactus. You can still go up towards Waimea, and there are some around. But the plants and the biocontrol agents remain interacting. So we can go up there and still find the agent in some of the cactus. But now the cactus will be at a very low manageable level. So this is really the goal of biological control, where you keep your invasive plant at a low manageable level. So how does biological control happen? So first we have to choose the target. So you have to choose your target weed, and that can happen from scientists, from government agencies, and importantly from the people. So people are working the land, they're working their farms, they're out there, they know what's a problem. They know it's a problem for their livestock, they know it's really hard to get rid of, they see their native species disappearing. They attend meetings, they email scientists, they email politicians and say, we think that these plants are really problematic. And so it's the confluence of the public and science that decide which plants are worth doing research for biocontrol that we think might be effective. So once we choose a target, then you have to go to the place where the invasive plant is from. So in this case, Himalayan raspberry is from China. So we decide to target that plant. We have to go to China or to India, and we're looking for extremely host-specific agents. It could be herbivores, it could be a fungus, it could be a parasite, a parasitoid rather. We're open-minded, but again, the condition is that it has to be impactful and it has to be host-specific. Once we have made an educated choice about which agents we're going to go forward with, then we can begin testing. So testing can happen in the native range, where both the insect and the plant exist outside in the field. And then testing can also happen in quarantine facilities. For example, we have one in Volcano here. And then after all of that happens, and after many years of searching and testing and permitting, once everything has been approved, then we can release and monitor our biocontrol agent. So this slide is really great to show that biocontrol is a lengthy process and involves much collaboration and a lot of exploration too. Sometimes we have to go to places that are pretty tedious. So, but it's fun. I'm not. Okay. So now to be more specific to what I'm eventually going to be talking about of Clidemia. So this is just a slide to show that we have a bunch of plants on Hawaii that are quite invasive, and they all belong to the family of melastomes. And we have made some progress with choosing new agents to release. But one thing that's really interesting about melastomes is that all of the melastomes on Hawaii are alien. And so this actually allows us a little bit more flexibility in what kind of agents we choose, because of course everything will still be host specific, but we know that as long as the agent only attacks plants within the melastome family, we'll be safe in Hawaii Island. I'll mention it now just for Clidemia that we have these two agents that are in the pipeline, but I'm going to be talking about them a lot more, not a lot more, but I'll be addressing them towards the end of the talk. So, the star of the show. The target plant here is Koster's curse, as many of you know it. I call it Clidemia hirta. It is a perennial shrub. It came to Hawaii in 1941 to Oahu, and then it spread to other islands in the 70s and later. It has white flowers and young green fruit turning purple when ripe. It is aggressively invasive. And part of the reason why is because it can exist in sunny, shady, wet to mesic pastures, forests, and forest edges. Importantly, though, it does not grow as much on the Kona side, so it definitely needs a certain level of moisture. It could also grow from 0 to 4,000 feet. The birds are the main disperser of the berries, but it's possible that pigs are also contributing to seed dispersal. And as I showed you in the first photo, it can form dense monotypic stands and of course displace native plants. So it's a huge problem. I've talked to so many people that have it in their yard. It's incredibly difficult to get rid of, and annoying, and all of that. So Clidemia is native to Central and South America, as you can see in the green circles, but then the yellow squares are all the places that it has invaded. I believe it was first dispersed from Costa Rica to Fiji in the late 1800s or early 1900s by seeds in coffee beans accidentally. So something as seemingly benign as that has created a huge invasion all over the Pacific Islands. So since 1953, in Hawaii, there have been several different biocontrol agents released to try to control Clidemia. I'm just going to highlight two here and then talk about the one I'm working on now. So one is this thrips species. And there's two things that I want to say about this. One is that the insect itself occurred in sunny or partly sunny areas, but it did not affect flowers or fruit. But it could stunt and or kill plants at very high densities, but only in sunny areas. So wherever you have shade or forest canopy, the thrips did not do so well. And I was just reading about it, and it said that not until 1982 was post-release monitoring conducted. And I'll talk more about this in a minute too, but just to say that biocontrol, the interest for controlling certain plants can sort of ebb and flow and is really at the whim of a lot of public funding and support. And so sometimes we can go really far in a biocontrol program, getting so far as to release the agent, but then 10 years later, there might not be any funding to do follow-up. And I'm sure you can see why that's really important. So, you know, it was almost 30 years later that a large-scale post-release monitoring was conducted of this. So, and that's kind of what I'm doing right now with Clidemia. So I just wanted to justify why it is really important. So another one of the biocontrol agents is Poseidon, which is a leaf mining beetle. And if you can see my mouse here, so the larvae make this sort of mine in the leaves, and then as an adult, they can defoliate the leaves. And this is a great example because after the beetle was released, it was clear that there was negligible damage being done. So the beetle seemed to be established, although not at very high rates, but then it wasn't doing a lot of damage. And then it became known that it was being attacked by a parasitoid that was already present in Hawaii. And this is what we can call biotic interference. So there can be many reasons why biocontrol agents are not as effective as we wish they were. And in this case, one of the reasons could be that because a parasitoid that was already here started to attack it. So in any case, it has established, but it does pretty negligible damage. So then the star of the show, this is the biocontrol agent that I'm working on right now, which is Mompha trithalama. It was released in 1995. It lays its eggs on flower and fruit. Here, these are some eggs. The larvae develop inside the fruit, damaging seeds and preventing seed development. And when they're old enough, they emerge through an exit hole and pupate. So these are photos taken by my excellent technician, Rosalie, showing like very young first instar larvae all the way up to a larvae that's ready to pupate. This is an adult moth. They're quite small. The life cycle takes about 50 days, and you can find hundreds of eggs on one cluster of fruit, you know, containing 7 or 10 fruit. But the efficacy in Hawaii is really unknown. So how much damage, how much impact is this moth doing? How prevalent is the moth? These are all questions that we had. So again, I just wanted to say that the post-release monitoring is kind of what we're doing here, and it's essential to determine establishment and success rates, and it's not conducted as often as needed due to funding issues, again, the ebb and flow of public support. And another reason why we're doing it is because we want to release new agents, and it's important to compare the impact of different agents. So we would like to be able to say we're releasing this new agent and look at how effective it is compared to this other one that has been here for 20 years. So the goals of this project were first to determine the relationship between larval and fruit development. So we wanted to look at how larvae are developing along with the developing fruit. We wanted to quantify the establishment of Mompha along elevational gradients. So we have the opportunity here to look at the plant from anywhere from 0 to about 3,000 feet and see how establishment might differ. We also wanted to measure levels of parasitism. Again, this concept of biotic interference, are the moths being attacked? We wanted to look at if seasonality affects establishment at different elevations. And we wanted to see, we hope to assess the impact of the moth on seed production. So we haven't gotten to all these things yet, but these are all the big goals of this project. So here's a map of the east side of the Big Island. And the first thing that I want to draw your attention to is this Waiakea site. So if you're not so familiar, this first transect These sites here run up along Stainback Highway, which provides a really great elevational gradient for us. And here, this is the Waiakea site where there were some releases made of the moth. And basically at this site, we collected fruit of all different stages and then looked inside to see how the larvae was developing in certain stages. And that might be a little bit to handle. I will explain it really clearly on the next slide. But just to know that we have two experiments here. So the first one was just at Waiakea, where we looked at fruit development and larval development. And then the second experiment that we have here is where we have 3 transects. So again, this is Steenbeck Highway here. This is the Volcano Highway here. And this is Lapahoihoi. You can see that the distance between sites is much shorter. That's because the elevational gradient is much higher. At all of these transects, we collected fruit from only one stage and just looked at establishment of the moth and the presence of parasites. So part of the reason why I was invited to give this talk is because some of our research sites are within the Lapahoihoi Unit Research Access Map, access area, and then Natural Area Reserve here. So part of our work is within the HETF land, and we very much appreciate being granted access to look at the Clidemia plants in that area. So again, as I mentioned, we had this one site at Waiakea, and at this one site, we looked at the different stages of fruit development. So here we have everything from the young flower bud to a young flower bud with the corolla showing, petals open, small green fruit, big green fruit with purple, and then the ripest fruit at the end. And we collected about 30 each of each fruit stage, and then we dissected them and we looked for the presence of a larva, and then if there was a larva, we would measure the head capsule. And then in general, we were able to quantify whether there was ever a larva in the in the fruit. So even if we didn't find one, we have other ways to tell whether there had been one inside, mostly damage or the presence of an exit hole. So here are some of our results. So the first thing that we found is that as fruit diameter increases, so does the head capsule width of the larvae. This seems really intuitive. The fruit gets bigger, so do the larvae. But one thing that's really important to know about this is that we didn't really know a lot about the biology of the larvae once they were released in the field and the life history. So we didn't know if perhaps the caterpillars were growing to a certain point and then leaving their fruit and then going into another fruit. It seems that they're not. It seems that they really do develop alongside the fruit in a quite linear fashion. So the really cool results are these. So here again on the x-axis, we have all of the different fruit stages. And on the y-axis, we have the percentage of larvae, the percentage of the fruit that contained a larvae. And orange means that there was, and blue means that there was none. You can see here that there's some variation, but about 20% of any out of the 30 of each of these fruits that we picked were infested with an actual larvae. So when we dissected it, we found a larvae, or one emerged by the time we looked at it, except for the big ripe purple fruit. So the big ripe purple fruit, out of all of the ones that we looked at, there were no larvae inside. And so one hypothesis was that maybe fruit don't develop and ripen if they were infested by a larva. But we found that was not the case. So we were able, again, based on damage or based on the presence of an exit hole, we were able to determine whether or not a Mompha had been inside the fruit at all. And you can see that when we look at whether or not there was an insect inside the fruit at all, the numbers come down quite a bit. And here, about 75% of all the ripe purple fruit we found showed evidence of a larva inside, usually by an exit hole, but also sometimes just by damage. So it does seem that there is a very high establishment of the larvae inside of the insect inside of these fruits. and that the fruit can develop and ripen even if it has been attacked by a Mompha. So for the second experiments that we did, again, these transects, where we have 6 or 7 sites all the way from about 200 feet in elevation all the way up to about 3,000 feet. At each site, we collected 50 large green fruit per site, and we measured the fruit diameter. And we observed them for 1 week and then dissected them again for evidence of larvae and or parasites. So here's what we found here. So transect 1, this is Steinbach highway for those of you who are familiar, and it's the same pattern. So blue means that we did not find any larvae, and yes means that there were. And here is an increase in elevation gradient, and Y is on the y-axis you have the percentage. So here you can see that up until, at 18, almost 1900 feet, about half of the fruit had evidence of Mompha, but at the higher elevations, there was very little, if any, presence of Mompha. So we wanted to repeat this. Replication is very important in science. So we did this on the second transect, which is the volcano highway, and then also up at Lapahoihoi. And you can see that at the highest elevations, we find virtually none, no Mompha. So we go from huge rates of establishment to, more than 80% of all the fruit that we pick, we can find an insect inside, to virtually none at the higher elevations. So there could be a lot of reasons for that, right? It's higher elevation, it's colder, maybe it gets less rain, maybe there's something different about soil composition. So one of the things that we were able to look at was fruit diameter. And so here, blue is transect 1, which again is Steinbach highway, orange is volcano highway, and gray is Lapahoihoi. And you can see here that at the highest elevations, there's a significant drop off in the fruit diameter. And we could tell that in the field. When we were out collecting, we could tell as soon as you got up higher, the fruit just were so much smaller. Now, we don't know yet if the small fruit are prohibitive for the Mompha, or if there's other reasons why they won't go up to the higher elevations. We don't know yet. That warrants further investigation. We do know that there's no Mompha up there, well, virtually no Mompha up there, and that fruit diameter is quite small. So then just a little note about parasitism. Here we had the 3 different transects, different sites and different elevations. The point here is you're like, well, there's a lot missing here. Well, that's because we hardly found any parasites. So essentially the point of this small table is to show you that we sampled at 20 different sites over 3 different transects, and we found 5 total parasites. It's a small wasp, and it seems to attack the larvae, but there's very low rates of parasitism here, and we didn't find any in the second transect. Now, I do want to say that there's a caveat with these results, in that we want to design a better experiment that will actually target parasites. So here we were more targeting presence, absence of Mompha at the different elevations and seeing if we could find parasites. Now that we know more about, or parasitoids, now that we know a bit more about the parasitoids, what kind they are, and how they might develop, I think we can design an experiment that will help us really isolate and look at just parasitism. So there are parasites that exist, but they don't seem to have a big impact because they're occurring at very low rates. So, so to kind of start wrapping it up, I hope you're convinced that Mompha is well established across the east side of Hawaii Island in all fruit stages, except at higher elevations, and that we found the same pattern at all 3 transects. So once you get up, I mean, there is some variation. I don't know the exact cutoff, but it seems to be around 2,000 feet, 2,200 feet, where we see a complete drop off in the amount of Mompha that we find. A potential reason why they might not go up higher is because the fruit are smaller at higher elevations, but it's more likely that there's lots of abiotic factors contributing to the differences in fruit phenology that are not optimal for the insect either. And parasitism does occur, but for right now, all we know is that it occurs at very low rates. So what do we still have to do then? Well, we still want to assess the impact of the moth on seed production. So again, we're finding super high levels of infestation of the moth in all of these fruit, but we have no real concept of the level of impact that it's having. People still see Clidemia everywhere. Is it really making a difference in seed production? The next thing we want to do is repeat this entire experiment in September to determine if seasonality affects establishment at different elevations. So one thing that could surely be preventing the moth from going to higher elevations is temperature, and it gets warmest, you know, August, September, and even a little bit of October here. So we were thinking of, we want to do this again to see if with warmer temperatures, the moth decides to go to higher elevations. And then, as I mentioned, we want to conduct an experiment explicitly on parasitism to really quantify levels of that in the field. So then I touched on this briefly before. We do have 2 agents still in the pipeline. One is this nematode species that makes galls and prevents the production of the fruit almost entirely. So that seems like it could be promising. I'm pretty excited about a gall wasp that is also in quarantine. So you can see here that the wasp is ovipositing into a young fruit bud. Galling insects in general are known to be very host specific. I mean, to the point where they can make galls on their host species, but even if sometimes it's so specific that if the plant isn't the right genotype, they won't be able to make the right galls. So any galling insect I'm pretty interested in. There's such a long history, a long evolutionary history there that allows for the insect to even make galls and for the plant to respond in the way it does. I find them really interesting. So we do have these 2 agents that we're working on to hopefully in the next 2 years or so. I'm just hoping we might start to release here against Clidemia. So with that, I will say mahalo to the many partners that share a vision for conservation in the Pacific Islands. My supervisor is Tracy Johnson, and Rosalie Nelson is the one who did most of the work. She did all the dissecting, measuring head capsules, and the really awesome photos. She deserves props for that. And to the rest of the team. And with that, I'll leave it open to questions.
Thank you so much for that awesome presentation, Ellyn. Yeah, if you guys have any questions for Ellyn and the work that she is a part of, please either unmute yourselves and turn on your video and ask some questions, or feel free to type them in the chat. And we'll sit here for a few minutes and let you guys ask questions. Oh, there is a comment in the chat already. Nick says, Clidemia is well established in the Central Kona region.
Okay. Yeah, I admittedly don't do too much work over there. But I know, as far as I know, it's not as prevalent on that side of the island as it is over here. But maybe I should check my records more.
All right. Scott Van de Verg says, Mahalo, Ellyn. Have you observed large seed counts in the mature fruits you collected?
Yeah, so we haven't really quantified that yet. There's definitely seeds in the mature fruits. It's not that the moth is not preventing the production of all of the seeds, but how many, we don't know. That's the next thing on our list, is to really figure out how to quantify exactly how many seeds are being produced. We also would like to figure out if the presence of the insect is affecting how many how many fruits actually develop. So that could be another way. But it doesn't seem like that's going to be the dominant force. It seems like the presence of the insect inside the fruit will have some impact on seed production. And then I see the other question.
Yeah.
So yeah, this was, somebody asked about the first of the two agents. So yeah, this is a nematode species. I don't know. Tiddlencus. I haven't worked with that one at all, but my boss seems pretty excited about it.
Susan Cordell and Aaron Bishop say thanks.
Yep. My pleasure.
Hi, I was wondering if I could ask a question.
Of course, go ahead.
I'll turn on my video so you can see me, if you can. Hi, Chuck Camara. Thanks for the great talk. This is a more general question about biocontrol agents and host specificity in general. Given that there are no native mellostomes to the Hawaiian Islands, I was just wondering how critical host specificity is when testing agents on a weedy plant like Clidemia, if there are non-target effects found on non-native species in the mellostome family.
Yeah, so I can sort of say from my, how do I want to say this appropriately? It's a very good question, and I did sort of touch on it, so it's fair. I still think that any host testing that we do is going to we are still going to give all of the insects a battery of tests with plenty of different species that even if we don't suspect that they would attack, we would still want to make sure that they're very host specific. If there is some leeway, if we're very confident that, you know, Myconia, for like the moth will attack Myconia and Clidemia, but nothing else, then I suppose that's okay too. But for me, from an evolutionary perspective, if you are starting with an agent that has a larger host range and is not as host specific, to me, I'm not as comfortable with that. So even if, there's no, you know, all of the mellostomes that are on Hawaii Island are invasive anyway, or not invasive, but at least non-native, and we don't really care as much. I still don't think that anybody that I would work with has the attitude of, well, we don't have to worry as much. I still think host specificity is very important in biological control. So it is, I mean, on one end, you know, we have a little leeway and don't maybe have to worry so much in this case, but I still don't think that's our attitude really. It's just probably going to be really safe, and that's good. That's what we want. We want it to be really safe.
Thank you.
All right, so we have more questions. Jean asks, do moths feed on any other species of mellostome?
Well, I know that there is a closely related species that will attack Myconia, but it's not this species, but it's a closely related species. Also, I'm not going to get into it now, but what is a species? It's a really hard question, and there are closely related, like a Mompha complex, as it were, that some taxonomists are trying to say are different species. But it's not as straightforward as of an answer as I wish that I could give you in that case. So there are some closely related species that will attack Myconia, which is, yeah.
All right. T. Lee says, thank you for the interesting presentation.
Yeah. Just saw the comment from Nick.
Yeah.
That's enough to prove me wrong.
Yeah. But let's see. And then Aaron says, can you recommend where to find more on evolutionary theory and how it's incorporated into the groundwork? What factors may help determine where to do host testing in native region or here in Hawaii?
Yeah, so I can get you started at least, not to be self-publicizing or anything, but we did a review. I did a review with 3 of my coworkers last year. So if you type in my name and look up my publications, and it's a review on the use of evolutionary theory in biocontrol. And in there would be a lot of references into where evolutionary theory can come into all the different aspects of biological control, everywhere from choosing an agent to rearing an agent in quarantine in the lab, what happens to lab populations, and then also what we expect in terms of adaptation after we release an agent into the field. That's actually one of my main interests, is what happens after. You know, when we release these agents, they're under intense selection pressure. And it doesn't mean that they're going to evolve and start to eat almond trees. But that was always my go-to phrase in California. But we do need to be aware of what's happening with genetic variation and the potential for rapid evolution. So if you go to that review paper, I think that might be a great place to start. And if you can't access, I think it's open access, but if you can't find it, please feel free to email me, and I'd be happy to get that to you. Your second question, so host testing, that usually, so that's done, I want to say that's done in both places. So we can definitely do host testing in the native range. One problem with that is that sometimes the plants are different. So, you know, even if it's only been 100 years, there can definitely be genetic separation, and the plants might not necessarily be the same. But we do start in the native range. So, and it's easier, right? It's so much easier to do the testing in the native range because you can grow the plants outside, you can have the insects outside, instead of restrictions in the invasive range where you would have to do it in a very secure quarantine facility. So we would definitely do that kind of testing in the native range, but then when we do the further testing in quarantine, it's much more lab-based. So it's very structured. We know the exact plants. We're exposing the insect to plants in the introduced range, and it's more of a controlled environment. So I would say that, yeah, we definitely do this kind of testing in both the native and introduced range.
All right, so there is one more comment / question, and it is from Randy Bartlett, who says, Mahalo nui loa no ko kukua e Ellyn. Keep up the great work. We formed the Melastome Action Committee, the precursor to MISC, in 1991 to search for ways to control Clidemia hirta, Dibashina species, and Myconia clavicens on Maui. It's great to see the work continuing nearly 30 years later.
I didn't know that MISC used to be like Melastome Action Committee. That's kind of funny to me. Like that was the biggest problem. Good to know. My pleasure.
All right, if anybody has any more questions, feel free in the next minute or two to put it in the chat or to unmute and ask. But other than that, I think we're about to wrap up. So thank you all for tuning in to this webinar and for being here and supporting the work that's being done and supporting Ellyn, who was kind enough to give us her time and share what she's working on with us today.
Well, thanks for having me.
Of course. Yeah, so thank you guys for tuning in. Thank you for being here, and I hope you enjoy the rest of your day. We will be having another one of these webinars in September 9th. We will be having our next webinar be about Hawaiian hoary bat conservation. So keep an eye out for that. We will send something out to everybody who tuned in today via the Eventbrite. And yeah, we hope to continue to see you guys. Oh, and also we'll send out the feedback form. So please fill that out and tell us what you thought about the webinar and if you liked it, if you have any ideas for improving it. All right. Well, everybody have a wonderful day.
Bye. Thank you.